This conference will now be recorded.
Fantastic. Well, welcome and hello. It's great to see so many of you here today. Marg, as we know, it's 2 o'clock in the morning in Australia, so thank you so much for joining us, and to everyone else who's joined us live. It's fantastic to see so many friendly faces. And before I go any further, as I just said, the webinar will be recorded. So if you if you prefer not to be seen on screen, then please do turn your cameras off. We're really excited about launching this new webinar series for our members. We, as you know, we're going to be inviting expert speakers to discuss a range of topics that will be of interest to all of you, we hope. If there are topics or speakers that you'd like to see included, please do let us know, and we will endeavour to arrange that. The next members webinar will be held during the ASIF Council meeting. And these 45-minute sessions are designed to be interactive. So please do think about your questions for Dr. Brown, and you're welcome to either pop them into the chat or at the end to ask them live. Just a few little housekeeping rules. If you could mute yourself during the presentation just to avoid any feedback. For those of you who have just joined us, if you could say hello in the chat, state your name and where you're from. It's great to get an idea of who's here. And if we do have any trouble with the connection, we might ask you to turn your cameras off. But otherwise, if you're happy to keep your cameras on, that would be also great. Following Dr. Brown's presentation, as I said, there will be the chance to ask questions, and we can have a discussion. So if you don't want to wait until the end, you want to put your questions into the chat, please feel free to do so. Well, we're delighted to welcome Dr. Brown to our webinar. I know that some of you do know Dr. Brown, having worked with him previously, but for those of you who haven't, Matt, I'm just going to say a few words about you. Matt's a clinician scientist who trained initially in medicine and rheumatology in Sydney, Australia, before completing a doctorate of medicine based at the University of Oxford, focusing on genetics of ankylosing spondylitis. He was appointed a Professor of Musculoskeletal Sciences at the University of Oxford in the UK in 2004. In 2005, Matt returned to Australia, firstly to the University of Queensland, and since 2016 at Queensland University of Technology, where he was the Professor and Director of Genomics. In 2013, he was elected to the Fellowship of the Australian Academy of Sciences in recognition for his achievements in genetic research. In 2019, he moved to King's College London, King's College London and to Guy's and St Thomas's Hospital NHS Trust to direct their NIHR Biomedical Research Centre. And in 2021, he moved to the position of Chief Scientific Officer of Genomics England. Dr. Brown continues to work in genetics of human diseases with a particular focus on common and rare bone and joint diseases, and in cancer genomics, as well as personalised medicine. He continues to practice rheumatology with a particular focus on spondyloarthritis. Today, we are really excited to hear from him about his research on the potential to treat axSpA via the gut microbiome. Welcome, Dr. Brown, and thank you so much for joining us today. I'm going to, bear with me. I'm going to stop sharing my screen. And Dr. Brown, if you would like to share your screen, I'm just going to give you the access. Bear with me.
So I guess I should start by apologising for the dreadful photograph that you were all just shown, but I'm not sure. I'll have to get a different one taken, I think, apart from that one. I was obviously in a grump when that was taken. Look, Joanna, thank you very much for the opportunity to present today. I think this is a really good initiative, and I hope that we get some lively discussion after I've given my presentation. So I'm going to talk for around 15 to 20 minutes, and then basically I'm happy to take questions for however long it might be that you all gone to sleep, and that you don't have any questions, but hopefully this will actually be stimulating for you, because I think it's hopefully what I'll convince you from this is that the gut actually, or gut-based treatments actually give us a real chance of actually doing something completely novel that might make a difference to the natural history of axial spondyloarthritis and ankylosing spondylitis. Now, can I just ask, is my screen actually sharing fully, or is it actually just in are you seeing it by the single slide?
Just in the single slide at the moment. It's not showing as a
Yeah.
as the slideshow.
Okay. I'm trying to avoid turning off the getting out of the presentation altogether. So here we go. How about that? Is that better?
Well, yes, we can see it as the slideshow, potentially with your notes if you have notes as well, rather than it's showing that screen.
Oh, I wonder how I've managed to get to that end slideshow. So just bear with me a second. I should be able to. We should be able to get to.
It's the display settings on the dark screen.
On the dark
it's
screen.
Showing your second screen.
That
I'm
one?
taking it you've got two screens.
Yep.
So on the other screen where we could just see the notes,
Yes, that's
right at the top, you've got display settings, and you can choose the other screen.
I've got slideshow, but I don't see.
Yeah, I think you have to start the slideshow, and then that black screen that we were just looking at will come up again. And then the display settings at the top there.
There you go.
That one.
And then swap presenter view and slideshow, I think.
How's that?
Wonderful.
Perfect.
Okay, fantastic. All right, thank you for that help. Okay, so, all right, so first up, I'm going to talk about what the evidence is that AS is actually a gut-driven disease. And then I'm going to talk about what evidence there is so far from studies, which is going to be a very short presentation section. And then talk with you about a clinical trial which Versus Arthritis has funded in the UK, and which we're shortly going to kick off, of a trial of fecal microbiome transplant in axial spondyloarthritis. So I think if you go back in the literature, people have been hypothesizing for 40 or 50 years that ankylosing spondylitis is a gut-driven disease. But the level of evidence that that is the case has increased considerably in recent years. And I'm going to show you data from human migration studies, from genetic studies, from studies looking at the bacterial profile that you find in the stool and gut of patients with ankylosing spondylitis, a little bit about animal studies, and then I'll talk about the therapeutic trials. Okay, so I think the strongest evidence is actually that AS is a gut-driven disease comes from the disease reactive arthritis that many of you will know is a form of arthritis that's related to ankylosing spondylitis, in which about 10% of patients who get affected with this condition go on to develop ankylosing spondylitis, and where the arthritis is known to be triggered by bacterial infections either in the urinary tract or in the gut. And so the fact that this actually does in roughly 1 in 10 patients actually go on to cause a disease which is indistinguishable clinically from ankylosing spondylitis indicates that at least a proportion of AS cases are due to bacterial arthritis. So we also know that ankylosing spondylitis commonly co-occurs with bacterial-driven gut inflammatory diseases, particularly Crohn's disease and ulcerative colitis. And so inflammatory bowel disease is definitely thought to be caused by bacteria driving inflammation in the wall of the gut. And roughly 2/3 of ankylosing spondylitis patients have inflammation in the small intestine, which resembles Crohn's disease, but is nowhere near as severe as Crohn's disease. and correlates with disease activity in ankylosing spondylitis, suggesting that it probably has a causative role in the disease. And then I try to stay clear of animals because animal model data often is animal models are not the same as humans, but there are some good animal model data, and in particular Joel Tarog's HLA-B27 transgenic rat model, where this rat, which has large numbers of copies of HLA-B27, remains free of arthritis if it's brought up in a germ-free environment, but the rat develops a spinal and pelvic arthritis, as well as skin condition like psoriasis, that if it is exposed to even simple bacteria. And the arthritis in the model can be prevented by using a probiotic, Lactobacillus rhamnosus, suggesting a potential for therapy in human disease. Okay, so this is about migration studies and looking at people with HLA-B27 living in different environments, and that's a much younger me from the mid-1990s, showing people from these kids who are going to school in an inland sub-Saharan area of the Gambia in West Africa, where I'd come from, which was Sydney. And basically, what I was doing here was that it was known that ankylosing spondylitis is very rare in people of African ancestry, and that for the majority of them, the reason that's the case is because HLA-B27 is very rare in Africa. However, in one small area of West Africa in the Gambia, there is actually a high prevalence of HLA-B27, very similar to what's found in European populations. And so I did a survey of people carrying HLA-B27 in this country, and found that basically they don't get ankylosing spondylitis. And so far since that time, now, how long, 27 years ago that I did this study, there still hasn't been a case of ankylosing spondylitis reported from that region, suggesting that there's something protecting HLA-B27 carriers in that region from developing the disease. However, this was an area that was heavily affected by the slave trade in earlier times, and a lot of descendants of people from this area in West Africa now live in developed countries, particularly in North America, where it has been shown that they do actually get ankylosing spondylitis. So you have people who are genetically identical but in different environments. One group gets ankylosing spondylitis and the other group doesn't, indicating that there's something about the environment that's protecting the people in West Africa or is triggering the disease in developed societies. And now it's interesting that there's a similar finding with regard to rheumatoid arthritis, and that in urbanized African populations, rheumatoid arthritis is becoming more common in those populations, suggesting that bacterial or something to do with the environment is actually making it, is also involved in rheumatoid arthritis. Okay, so most of my career I've spent my time trying to identify genes that are actually involved in ankylosing spondylitis. You'd probably all be aware of the fact that HLA-B27 is involved in the disease. It's the major gene for the disease, and it is responsible for about 20% of the total inheritance of ankylosing spondylitis. But so far we've identified another 116 other genetic variants which are definitely involved in the disease, robustly so. And we don't know how all of them actually cause the disease, but we know what tissues they're expressed in. And so looking at the tissues that those genes are expressed can tell us, well, you know, basically if they're involved in the disease, they have to be expressed in the tissues. They have to be expressed in particular tissues, and the tissues that they're expressed in tells you about what tissues are actually involved in driving ankylosing spondylitis. So we looked across the different cell and tissue types for which there was gene expression data, and what you can see here is that the height of each of these bars represents how much more often than by chance the AS-associated genes are actually expressed in those tissues. So not surprisingly, the AS genes are expressed in lots of immunological cells, particularly T cells, which we know are involved in ankylosing spondylitis. But actually the most strongly involved tissue is gastrointestinal tissue, indicating that the genes that increase the risk of ankylosing spondylitis are largely expressed in gastrointestinal tissue, and therefore gastrointestinal tissue must be involved in driving the disease. Okay, okay, so mice, I think this is one of my last, or if not my last, mouse slide. This is a slide of a not very happy looking mouse whose ancestors were brought to a research laboratory in Brisbane that I was working at when I was doing clinics together with Mark Lewington. And it was imported by Professor Ranjani Thomas, who's an outstanding immunology researcher who focuses particularly on rheumatoid arthritis. And she imported this mouse, which is called this SKG mouse after its discoverer called Simon Sakaguchi. And it's supposed to be a model of rheumatoid arthritis, and she was going to use it for research in rheumatoid arthritis, because in the animal houses that this mouse species was discovered to develop arthritis, it was in Tokyo, and it was basically had arthritis affecting its paws, and was thought to have rheumatoid arthritis. Anyway, when it came to our mouse house in Brisbane in a different environment, it developed, you'll see in picture E, a hump. And it's a bit difficult to see, but in pictures C and D, it actually has a swollen right eye because it's got anterior uveitis. And when the hump was looked at closely, it's got a hump because it's got spondyloarthritis. It's got sausage digits like psoriatic arthritis, and its gut has inflammation in it with gaps between areas that are normal and areas which are thickened and inflamed, which closely resembles Crohn's disease. So basically what's happened is that a mouse in one animal house in Tokyo in a particular environment gets rheumatoid arthritis, and you change the environment and put it in a different environment, the same genes, and it gets a spondyloarthritis with spinal inflammation, anterior uveitis, inflammatory bowel disease, and a peripheral arthritis that looks just like the sort of peripheral arthritis that patients with seronegative diseases like axial spondyloarthritis and psoriatic arthritis gets. Okay, so humans are not mice. So what's the data that humans actually have different bacteria in their gut? So this is a study that we published in 2014, which was the first evidence that AS patients actually have different gut microbiome or different bacteria in their gut than people who don't have ankylosing spondylitis, healthy people. So what we did was we used a special new technique using genetic sequencing methodology to characterize the bacteria that were found in biopsies from the distal small bowel, the terminal ileum, of patients with ankylosing spondylitis compared with healthy controls. So this was a completely novel technique. In the past, we'd had to grow bacteria up in agarose plates and try and identify the bacteria from there and count them. But with the development of much improved DNA sequencing approaches, we could sequence all of the bacteria in the gut and identify what the bacteria were from the sequences and then count them very accurately. And so we then used a statistical method to look at the similarity of bacteria between a bacteria found in individuals with each other. And so each in the graph on the left, every dot represents one individual, and they've been profiled according to basically what bacteria they've got in the gut to see how similar they are to the other individuals. And the people with the red dots have all clustered together. They're the people with ankylosing spondylitis, whereas the people who had the blue dots are people who are healthy and don't have ankylosing spondylitis. And we could quite easily distinguish using the bacterial profile from the gut the patients who had ankylosing spondylitis and those that did not. And we found a bunch of bacteria that were found more often in AS patients, presumably risk bacteria for the disease, and a few bacterial families that were decreased in prevalence in ankylosing spondylitis, which are presumably protective species. So that data has been looked at, or that sort of study has been repeated multiple times since then, and has been replicated in cohorts from North America, from Europe, and from China and East Asia, indicating that there's basically no chance that it was a false positive. It's a definite finding. However, what it doesn't prove is whether the bacteria are causing ankylosing spondylitis or whether the ankylosing spondylitis itself is causing the change in the bacteria. So the classical chicken and egg issue. So to try and work that one out, what we did was we went and worked with a group from North America, led by a guy called Jim Rosenbaum that many of you will know from Oregon. And so he and an outstanding research fellow of his, who sadly passed away, called Mark Asquith, had collected a large number of bacterial and stool samples from patients who were having colonoscopies done for screening for colorectal cancer. So were basically over 50s people just wanting screening even though they had no symptoms. So these are patients who have no ankylosing spondylitis and in fact no other disease, who are healthy, who are just being screened. And so what we did was we collected samples from these individuals, genotyped them for HLA-B27, and then also sequenced their bacteria and found that in each of these clusters, so the blue dots in this one are people who are HLA-B27 negative, and the red dots are the people who are B27 positive. And using biopsies from the gut wall or from the stool, we could distinguish those who are HLA-B27 positive from those who are HLA-B27 negative, indicating that that HLA-B27 changes the bacteria in the gut wall and in the gut itself in the absence of any disease. So it's not the disease causing the change in the bacteria, it's the bacteria causing the disease. We also, in a Chinese study, went and looked, in this case, looked at patients who are AS patients who are either on biologics treatment, so TNF inhibitor treatment in this case, so they're the green diamonds, versus those who had AS but were on no treatment, who are the people with the blue crosses, versus healthy controls. And we were able to distinguish the healthy controls from people who were untreated, that is between the orange and the blue dots, but we could not distinguish between healthy controls and the AS patients who were treated, and we could easily distinguish between the treated AS patients and the untreated AS patients. So in other words, ankylosing spondylitis treatment, successful treatment with TNF inhibitors, normalizes the gut microbiome, suggesting that maybe one of the ways that TNF inhibitor treatment works is actually through the microbiome. So how might the microbiome actually cause ankylosing spondylitis? There are a few different theories about how this might occur, and it may be that actually all of these mechanisms are actually involved because there is evidence to support each of them. So one is that AS patients do have increased gut permeability. So this has long been known, and using a range of different methods, people have shown that the permeability of particularly the small intestine of AS patients and of their first degree relatives is increased compared with healthy controls. And so it's hypothesized that this may lead to increased movement of either whole bacteria or bacterial toxins from the gut into the circulation, leading to ankylosing spondylitis. So here's some evidence to show this does actually occur. This is a paper published by Francesco Ciccia, a really good Italian ankylosing spondylitis researcher. And so these are gut biopsies from ankylosing spondylitis, and the A, B, and C are from AS patients, and D is from a healthy control. And he stained it with a special sort of silver stain where the bacteria are stained black. And you can see in the AS patients where the red arrows are, there are bacteria in the gut wall actually penetrating the gut wall and progressing to migrate into the submucosal area of the gut in the AS patients, but there are none in the ankylosing spondylitis patients. So they then counted up those bacteria and counted up the number of inflammatory cells in the gut, and the number of bacteria present in the gut actually correlates very well with the amount of inflammation in the gut in those patients. Okay, so then the third thing they did was that they took serum from these people, and the bacteria have in their gut, in the bacterial cell wall, something called lipopolysaccharide. It's absolutely characteristic of bacteria, and it drives inflammation in patients. It's a major cause of fever when you have a bacterial infection. And so the AS patients were shown to have much higher levels of lipopolysaccharide in their blood than healthy controls. And what's particularly interesting about this is that one of the genes that is strongly associated with ankylosing spondylitis risk is the receptor for lipopolysaccharide. And so this lipopolysaccharide interacts with that gene to drive inflammation in ankylosing spondylitis. So this is, I think, with little doubt, definitely part of the reason that people get ankylosing spondylitis. Okay, the next one is a bit more complex. And that is that we looked at in the Chinese patients, there were groups of, as I mentioned before, groups of bacteria which are increased in prevalence in ankylosing spondylitis, and then groups of bacteria which have reduced prevalence in ankylosing spondylitis. And what we found is that some of the bacteria which are known to be anti-inflammatory or produce anti-inflammatory product were amongst those that had reduced prevalence in ankylosing spondylitis, and that when you treated those patients, the prevalence of those bacteria increased. So this is looking at a particular bacterium called the Calibacterium prausnitzii. I can pronounce it, but don't ask me to spell it twice accurately. And so this bacteria produces short-chain fatty acids that protect against inflammation. And in the untreated ankylosing spondylitis patients, their prevalence was reduced compared with healthy controls, whereas amongst treated ankylosing spondylitis patients, it had exactly the same prevalence. So AS is associated with a reduction in this protective bacteria, and that may be one mechanism by which AS is a bacteria-induced AS. All right, now the third one is a bit more complex, and this is a theory called molecular mimicry, where basically it was hypothesized for many decades that ankylosing spondylitis is caused because HLA-B27 may be presenting or reacting to what we call antigens, so foreign bits of the bacteria that resemble parts of the body that are found at entheses, so the parts that inflammation occurs in ankylosing spondylitis. And because the immune system is recognizing both as exactly the same way, when you get exposed to a bacteria, it drives inflammation which affects both immunity against the bacteria, but also inflammation at the site where that antigen is found in the body, that is the enthesis. And so there is now increasing evidence that this might be the case. And so this is, again, a paper that our group published from Brisbane 2 years ago. So HLA-B27 operates to present these foreign particles or antigens to the immune system, and part of the immune system lymphocytes called CD8 T cells. Now, in your body, you have literally over 1 billion different T cells, each of which has a receptor on it called the T cell receptor, and that T cell receptor is what recognizes this foreign antigen or foreign particle. So each of those billion different T cells has a different T cell receptor. And when the T cell actually recognizes a foreign particle or antigen, that T cell is stimulated to reproduce, and so you then end up with very large numbers of hundreds of thousands to millions of those T cells. And so if you see large collections of T cells expressing a particular T cell receptor, it means they're reacting to something in common. And what we found was that amongst ankylosing spondylitis patients, roughly half of the patients carried a very specific, an expansion of a very specific T cell type that carried a single T cell receptor or one very closely related to it, compared with only about 1 in 10 HLA-B27 positive healthy individuals. And this indicates that the AS patients are reacting to something, all of these AS patients are reacting to something similar to each other. And what was really exciting was that T cell type had actually been identified before. It had been identified in a patient with bacterial-induced reactive arthritis who'd had bacterial dysentery and had caused reactive arthritis. So these half of AS patients had exactly the same T cell as had previously been reported over 20 years before in a patient with bacterial-induced reactive arthritis. And that says that these patients, the AS patients, are almost certainly reacting against a gut bacteria. Anyway, so I think that this basically shows that AS is a gut bacterial-driven disease, and we're hoping to use the T-cell receptor to identify what the actual bacteria is that is causing the disease. But in between times, there are things that we can do, simple things, which might actually lead to a therapy for AS patients. I think it would be hard to remember a clinic of mine in AS where I haven't had a discussion where a patient's asked me about, is there anything that they can do for their ankylosing spondylitis, such as can they change their diet? So there's a huge amount of interest in this, and a lot of publications, in particular in recent years, looking at diet and ankylosing spondylitis. So although I've found 90 different publications with a simple search of diet and either ankylosing spondylitis or axial spondyloarthritis in the PubMed citation database, actually the number of actual clinical trials involved is very small. In fact, only one clinical paper so far reporting a clinical trial with an effect on ankylosing spondylitis of a dietary intervention. So pretty disappointing. These are the treatment options that you might consider if you were doing a study of trying to affect the gut microbiome in ankylosing spondylitis. So the clinical trial was actually a trial of short-chain fatty acids. This is the dietary trial, and it basically showed no beneficial effect. Whether they were using the right dietary intervention or not is not really clear, so I think that one's basically an early but not imperfect study and doesn't really help us that much in deciding whether gut-targeted therapies are going to be beneficial or not. There are no other formal studies of diet in ankylosing spondylitis, despite the extensive interest that patients have in diet and the fact that I think the vast majority of the patients that I see have tried dietary interventions of one variety or another. Okay, so probiotics and prebiotics are two other ways that people have thought about. There is one, so probiotics are actually, that consists of giving patients live microorganisms with the aim of changing the microbiome or the bacteria in the gut. And there's one trial of that, which is a trial from New Zealand from Simon Stebbing's group. And they tried that, but basically showed no beneficial effect. It's not clear in the study whether the probiotic actually made any difference to the gut bacteria, and so we don't know whether the treatment in itself actually affected the gut bacteria, and therefore it's really unclear why the trial actually studied. So prebiotics are, that's giving agents which induce changes in the gut flora but which are not bacteria themselves, and there are no published trials of that in ankylosing spondylitis. There's no trial of bacterial metabolites. There are no trials of antibiotics. This is a bit of a surprise because antibiotics have been trialed in reactive arthritis with some beneficial effects in some studies and not in others, but no trials in ankylosing spondylitis. There have been really good studies in inflammatory bowel disease, and some of you may be aware that actually antibiotics are used in both pediatric inflammatory bowel disease with quite marked effects, and in adult inflammatory bowel disease with, it's pretty much standard of care for severe inflammatory bowel disease to give antibiotics like metronidazole. And then finally, fecal transplant therapy, which I'll talk about next. So fecal transplant therapy consists of literally trying to recolonize the gut with healthy bacteria by giving transplants of healthy fecal material from healthy donors. Usually, this has been given by obtaining fecal samples, which are then, after the patient has had a washout of their colon, they have a colonoscopy or an enema, and the transplant is delivered to the large bowel and the distal small bowel. However, there are, in recent years, people have developed alternate methods of giving this therapy by what's called lyophilizing or freeze drying the bacteria and then encapsulating them, putting them in capsules and asking the patient to swallow the capsule. The capsule goes through the stomach. It's protected, enteric coated, so it doesn't get dissolved in the stomach, and then basically releases in the small intestine, and that results in bacterial delivery to the intestine in that way. These sorts of treatments have been trialled and been shown to be effective in inflammatory bowel disease in gold standard trials, so randomized placebo-controlled studies in inflammatory bowel disease, and shown to be result in quite significant remission rates. So basically, this is looking at steroid-free clinical remission and endoscopy remission or response found in a quarter of patients on treatment versus only 8% on placebo, statistically significant, and a whole bunch of secondary outcomes also statistically significant, except for steroid-free endoscopic remission, but the others were did show basically significant responses with very few adverse reactions, largely some mild abdominal discomfort, some flatulence, and that's about all. There have been a couple of other trials which have looked at this. Some have been positive, some have been negative, but basically it's now fairly accepted that this treatment does work for inflammatory bowel disease. So with that in mind, we have applied for and obtained funding to run a clinical trial of fecal microbiome transplant in axial spondyloarthritis. It's both a safety and an early efficacy study, so what's called a phase 1 and 2 clinical trial. We need to demonstrate safety because of the fact that we know that bacterial infection can induce ankylosing spondylitis. We therefore need to be sure that fecal microbiome transplant doesn't also exacerbate it or induce it, although to date, there are no examples of ankylosing spondylitis occurring as a complication of fecal microbiome transplant in inflammatory bowel disease or in any other indication. So I think it's that we can be quite confident that it will be safe, but we need to be sure, and that's why it's being done as a phase 1/2 study as well. So we're looking at patients with very early disease because we want to be sure that we're seeing patients with active inflammation and where pain is being caused by inflammation, rather than patients who've got more long-standing disease where they may be having pain because of ankylosing spondylitis induced damage, in which case an anti-inflammatory treatment is not going to benefit them so much. So we're looking at patients who've got non-radiographic axial spondyloarthritis who have objective evidence of inflammation both by MRI and by having an elevated CRP, and who have significant pain. And of course, we've got some exclusion criteria. We're not giving it to patients who've had antibiotics in the previous 3 months because obviously that will change their gut microbiome anyway. We're not giving it to patients who've been treated with sulfasalazine, which we also know changes the gut microbiome, or TNF inhibitors. Similarly, prednisone, pregnancy, known inflammatory bowel disease, or other major diseases, and the inability to swallow capsules are also exclusion criteria. The patients are going to receive 2 treatments at baseline, one at baseline and one at 2 months, where they swallow a bunch of capsules. So no need for colonoscopy in this case because it's the encapsulated version of fecal microbiome transplant. We're going to treat 30 patients and have 15 patients on placebo. We're going to watch for 6 months. And the main outcome measure is actually, because it's a safety trial, we want to see the safety and tolerability of the treatment, but we also want to see, did the bacteria given by mouth actually result in normalization of the gut microbiome? How long did the bacteria actually survive in the gut microbiome before it ultimately reverted to the pre-treatment microbiome? Was there any beneficial effect as measured by any of the standard regime measurements like the ASDAS, the BASDI, and also we're going to look at blood test response by CRP and ESR, and we're going to do MRIs at the beginning and end to see if we can see any difference in the MRI evidence of inflammation. So that study was funded last year. Unfortunately, of course, that funding had occurred at the same time as we were all going through the dreadful pandemic. And so we've not been able to actually get to start the study, but I'm expecting that this study will start in Guy's and St Thomas's Hospital and King's College Hospitals in London in the back half of this year. It will take us 2 years to complete, but if it's effective, then basically the plan would be to then run a phase 2 or phase 3 study and to look at a more diverse group of patients, including patients with established ankylosing spondylitis. I'm not aware of any other similar study ongoing worldwide, but this is an idea that I'm sure others will have. So hopefully other people will be doing similar studies given time. All right, so I'll stop there and hopefully go back to seeing everybody else on the screen. And I'm happy to take questions.
Fantastic. Dr. Brown, thank you so much. That was absolutely fascinating and really exciting with the clinical trial. I'm sure everyone will agree. We've had a few questions sent through already. Maybe if we start with that, and if others would like to pop their questions into the chat or pop your camera on, and we can come to you directly. So the first question that we've had is it possible to create a microbiome in the intestine and use it to reduce the activity of axial and/or psoriatic spondyloarthritis?
Just the first bit of it again, is it possible to
create a microbiome in the intestine and use it to reduce the activity of axial or psoriatic spondyloarthritis?
Yeah. Okay, so look, that's what we're trying to do with the fecal microbiome transplant, basically, is to replace the microbiome that the patients have, which is likely to be abnormal, with a normal one, and hopefully thereby benefit their arthritis. This has been trialed in peripheral psoriatic arthritis. There's been a study published in the last 12 months looking at peripheral psoriatic arthritis, which I think is a bit of a different disease and may actually be driven by a different microbiome. It may be driven by the skin microbiome because it's a skin disease particularly. So we'll see. But that's the goal of our FMT study.
Right, thank you. Zoe has said, really fascinating, thank you. Is there anything we could advise patients who ask about diet/gut treatments?
Look, I think the issue is that there is no evidence base for any dietary intervention. Now, all of the patients that I see who've tried diets are coming to see me because their arthritis is active. And so therefore, I would always tell you that diet doesn't work for ankylosing spondylitis, but I fully accept that that's an ascertainment issue because they're coming to see me in the first place because their diet hasn't worked. And it could be that there are a stack of patients out there who are not coming to see me where the diet has worked. But I think basically the recommendation I generally give people is that just actually following a normal, healthy, diverse Mediterranean style diet is probably the most sensible, but we'd have to say that there is no clinical trial evidence to say that any dietary intervention makes any difference at this point.
Okay, thank you. Michael has asked, and forgive me because I'm probably going to pronounce this wrong, so Klebsiella doesn't play much part in axSpA, is what he's asked, is that right?
Yeah, so this goes back to a story that's been going in the literature for 30 or 40 years about whether Klebsiella is a key driver of ankylosing spondylitis that originally originates from some studies using non-sequencing based methods, but particularly antibodies, suggesting that Klebsiella carriage was increased in patients with ankylosing spondylitis. These studies were done at a time when the ability to profile the bacteria in the gut was pretty limited. And now we have these really good DNA sequencing based methods, which are highly accurate and comprehensive. We know that Klebsiella definitely is not different in prevalence in ankylosing spondylitis patients and healthy controls. So I think that story now can be put to rest, and that Klebsiella definitely is not involved in driving ankylosing spondylitis. Also, there was a suggestion that low starch diets reduced prevalence of Klebsiella and that was beneficial for ankylosing spondylitis. But when you look back through the literature, there is no published clinical trial data to show that low starch diets benefit patients with ankylosing spondylitis, and therefore I don't think they should be recommended.
Okay, thank you. Another question we've had is, from what age can the microbiome assisted treatments be applied? Are you suggesting?
Yeah, so look, I think that's a really interesting opportunity for us, because this is a treatment which has no side effects effectively. And so if we can show that this actually is beneficial, and then a next step would be to see whether it's preventative in patients who have high risk of getting disease. Since we know that ankylosing spondylitis is highly heritable, it's over 90% inherited, we should be able to, if that's the case, identify high-risk individuals and then treat them with a preventative thing and prevent the development of axial spondyloarthritis. At the moment, that's really just a research theory. We've got to demonstrate that this actually works first, but it's a really exciting possibility.
Yeah, that sounds great. Thank you. Joe has asked, which bacteria are protective against gut permeability?
Okay, so we don't know what bacterial species are, but we know some families of bacteria like Prevotella vallinella, and the F. praunitzii is one species that we know are protective. But it's a long way from saying that these are, well, we know that they're not found as much in patients with active disease, and that when you treat the disease, they normalize. What we don't know is whether if you give these bacteria that they, or increase their prevalence by some mechanism, whether that protects and improves the gut permeability issue and protects against ankylosing spondylitis, and that's the next step.
Okay, thank you. Marg is obviously still awake, which is amazing considering the time in the morning.
Yes.
And she has just asked, is there a difference with the HLA B27 neg, and also can it help with diagnosis?
Okay, so Marg, as ever, asking really good questions. So the answer is there just haven't been enough HLA B27 negative patients studied. And so we simply don't know what the gut microbiome is like in HLA B27 negative ankylosing spondylitis. It's my bet is that it's actually going to be something different in the B27 negatives that's driving the disease, but we just don't have data on that yet. And then the next bit is about can it help with the diagnosis? So I'm trying to, if I've understood this correctly, the question is, can you use the microbiome to help with diagnosis? And the answer to that is yes. But multiple studies have now shown that you can clearly distinguish AS cases from healthy controls using a stool sample. And that's quite a possible thing. It's actually cheaper to profile a stool than it is to do an MRI scan. So actually, I can see this as a potential future diagnostic test. I guess the difficulty is that it's likely that the diagnostic test will be different in each country because of differences in diet. And so it may be difficult to standardize. So maybe that's why it hasn't really taken off yet.
Okay, thank you. Got lots of questions coming in, which is exciting. Miranda in South Africa has said, you talked about your work in West Africa and certain environments protecting patients with axSpA. Do we know what the environments are that it's protecting?
I wish I did. Miranda, that's the multi-million-dollar question, I guess. We just don't know.
Okay, thank you. Another question we've had is, how can we protect the gastrointestinal tract with the constant intake of non-steroid anti-inflammatory drugs?
How can we protect the gastrointestinal tract from the
constant intake of non-steroid anti-inflammatory drugs?
Okay, so look, minimizing the dose and using short-acting anti-inflammatories, I think, are really good ways of doing it. I don't think there's any way that you can fully protect the GIT from effects of non-steroidal anti-inflammatory drugs, but whether the changes that anti-inflammatories induce, obviously they induce the gastric and duodenal ulceration, which we can protect against using drugs like the proton pump inhibitors. They also have effects further down the intestine. What effects those have, what clinical effect that actually has, having inflammation further down the small intestine from using particularly long-acting anti-inflammatories, is not really clear at this point. So it may be that it's a problem that doesn't need a solution.
Okay, thank you. Michael had a comment going on from Miranda's question. He said he grew up in Gambia, so what did he miss there from protecting him from axSpA? He's HLA-B27 positive.
So I'm presuming that this means that Michael has ankylosing spondylitis, and I'm going to guess that from the fact that he says he grew up in the Gambia, that he's actually not still living there. No. And so at some point now, he made a mistake and left. Look, ultimately somebody's going to get, the Gambia has got different environments as well. And so, large cities like Banjul, Basay, they are becoming much more urbanized, and it would be no surprise if people start getting ankylosing spondylitis now that the environment has changed. The area that I was doing the survey in was right at the most inland area of the Gambia, very much in rural townships, which were extremely basic. And so if you grew up in those, then I have no explanation for it other than maybe you left a bit early, and that therefore you've now picked up something, or you've lost the protective effect because you've changed environments.
Thank you. And Miranda's actually just asked another follow-up question, focusing on Africa again. Do you think it will differ from West Africa to the south in terms of the protection?
Look, so I'm not an expert on genetics in people from different regions of Africa. Obviously, they vary considerably, because even in the Gambia, it was only the fuller group with ancestral group within the Gambia who carry HLA-B27. So my understanding was, and correct me if I'm wrong, that in South Africans of African descent, that HLA-B27 is very rare. And so I think in South Africans, it's likely just the prevalence of HLA-B27, which means that the disease is rare in that ancestral group. But take that with a major grain of salt, because I'm not an expert on South African genomics.
Thank you. Rich from the US has said, is there a reason we couldn't do an HLA-B27 neg FM study?
Well, so in the FMT study that we're doing, we're not excluding people who are B27 negative. It's so basically we're just taking people who have early axSpA who definitely have inflammation, and I really don't care whether they're B27 negative or positive.
Okay, great, thank you. I'm aware that we're running short of time. We just have another question, which was, Alexis said he'd like to hear more practical recommendations in terms of are there any special tips for patients from the work that you've done already?
Look, as I was saying about diet, the problem is that there's been a heck of a lot of expert opinion, but virtually no data. And so I'm not going to make a recommendation to an educated group like this based on hearsay, which is where we're at, basically.
Yeah, fair enough, fair enough. I think, unless anyone else has any other questions, I think that's it. We've gone through a lot of questions and we're right on time, which is wonderful. So Dr. Brown, I just want to thank you so much for that really fascinating talk. It was just so interesting, and I think everyone would agree with me in saying that they've learned a lot. And this session will be available on the ASIF website, and I know that and I know a lot of people will be sharing it with their local communities. So thank you. And Joe's just put her camera on. I'm not sure if she wanted to say something.
Well, I was just going to say, to finish up, I hope that I get an invite back in 2.5 years' time or so, when we've completed our clinical trial, and we can talk about whether it worked or didn't work at that point and what the learnings were.
We would love that. That would be fantastic. Yes, please.
Okay. All right.
Joe, did you want to say anything before we finish?
No, just thank you. That was absolutely brilliant. Thank you very much.
Thanks for your outstanding questions, and also thank you for staying up, and for those of you who are on the wrong side of the planet until ridiculous hours.
And Dr. Brown, if you ever want anything from us or our community, feel free to contact us. I'm sure
Okay.
everyone would be very excited to be involved some way, shape or form.
Yeah, look, over the last 30 years that I've been doing ankylosing spondylitis research, the support of the patient organisations has been absolutely outstanding, and is why we've made such headway with the AS genomics. So thank you all already for the support that we've had so far.
Fantastic. Well, there's lots of thanks and appreciation coming in on the chat box. So, but thank you again, Dr. Brown, and we'll let you go.
All right. See you then, everybody.
Bye-bye.
Bye-bye, everyone. Thank you so much for joining us.
Bye-bye. Thank you.
Bye.
Bye. Great to see you all. Bye. Thanks so much. I'm going to stop recording. Thank you.